Hello, everybody. Welcome, welcome. We are so excited to get into College Biology 101 with you all. As folks start filtering into the Zoom room, go ahead and kind of get acclimated to your Zoom window. First and foremost, I'd like to direct you down to your chat bubble at the bottom of the Zoom window. If you click that, you'll be able to type chats into the webinar chat. This is how we are going to be monitoring questions today. So please put your questions in there. We have a big favor to ask when you ask us questions, which is please set that little to section. If you click that blue button, you'll be able to select everyone. And we ask that you chat your questions to everyone. That's because as we answer your questions, we don't want to look like we're talking to ourselves. So we want people to share the wealth of information, figure out what other people are wondering. It just helps us build that community a little bit. So as you all are settling in for today's webinar, go ahead and make sure that chat is set to everyone. And go ahead and let me know. Um, a fun thing that's happened in our home state of Oregon out here. Our Portland Zoo just had a new, I think, elephant baby last week, just before Valentine's Day. What is your take on the huge? I don't even know if it's like a push per se of baby animals at zoos, but holy cannoli, it started with that hippo in Thailand and it's just everywhere now. So what are your thoughts? Tell me your baby animal thoughts, which shouldn't be too hard to come by. I'm pro, by the way. Pro baby animals. Yes, Colleen says the more cute baby animals, the better. You are so right. What
I'm
about
obsessed with the baby tapir. That little, I don't know if you've seen that one. I think it might be Cincinnati Zoo. I can't remember which zoo it is because apparently I follow all of them.
I
It's
love that.
Washington. It's what Point Defiance.
So Point Defiance, that's what it is. Yes.
Oh,
It's
okay.
all, he has like stripes and spots. Super cute.
I'm Googling that as soon as we're done here. All right. And just to make the most of our time today, I will hop right into some housekeeping items. First and foremost, you will be getting a recording of today's webinar. It usually takes us about 3 to 5 business days to get that edited, but as soon as it's edited and live, you can find it on both our website and our YouTube channel. Once it's posted, I'm also going to email the recording to you. So keep an eye on your inbox. You will get both a link to the recording as well as a link to today's resources folder. In that resources folder, you'll find the presentation for today as well as some experiment files. I'm going to drop a link to that folder in the chat here as soon as I'm done. And as I mentioned before, we want to answer your questions during today's session. So please use that chat section. Make sure that to section is set to everyone so we can all see what questions are being asked. And if we happen to not get to your questions, please reach out to us at biology@vernier.com. I'm going to go ahead and launch a survey for you all to take, and I will drop that link in the chat in just a moment. So I'll pass it off to today's presenters, and thank you all.
All right. Thank you, Dylan. All right. So I'll just introduce you to the agenda. My name is Sara Tallarovic, and I am going to be one of your presenters today. I am joined by Colleen McDaniel, and we'll spend a little time to introduce ourselves in a moment. There is Dylan's survey. So if you could let us know, do you currently own and use Vernier? Yes or no? And secondly, how comfortable are you with Vernier products? That'll give us an idea of who is in our audience and how we can customize our discussion today. All right, so most of you have products and use them. Looks like one person doesn't yet. And for comfort level, we're kind of all over the map, and that's okay. That's why we're doing what we're doing. So, all right. All right, so we're going to do brief introductions, tell you a little bit about our background. And then also we will introduce you to both software and sensors. We're going to highlight kind of a key group of sensors, but we can also talk about others. And certainly if you have any questions, let us know, drop them into chat. I'll do a couple of quick demonstrations. So just to kind of show, especially for people who are newer or maybe haven't used our web-based app, we'll do some demos of that. And then obviously the chat function is up and working. So please drop any questions you have into chat. And we will discuss as we go along. So you can drop chat in at any time. So to introduce myself, I have a PhD in zoology from Oregon State University, and I taught for 15 years as a biology professor. I taught everything from like regular freshman bio, majors and non-majors. I taught human anatomy and physiology. I taught neuroscience courses. I taught endocrinology courses, also vertebrate zoology and animal behavior. So I was at a small liberal arts school, and I was a small portion of the biology department. And we had a little bit of everything. I also was a researcher in animal physiology and behavior, as well as neuroscience and behavioral endocrinology. And so that's my background. I joined Vernier because I started getting more and more into educational technology and faculty development, and really enjoyed working with faculty members. So, and then Colleen, would you like to introduce yourself?
Yeah. Hi, everybody. I am a biology and environmental science uh, specialist here at Vernier. I've been at Vernier for just over 11 years. Before that, I was actually working on a PhD, but dropped out to become a teacher instead, and much preferred talking to students than I did talking to fruit flies. Uh, I taught for 6 years in the Houston area, where I taught freshman biology and AP environmental science. And yeah.
And Colleen, you were also a lab manager, correct?
Oh, yes. Thank you for a reminder. When I spent a good amount of time in college as the assistant lab manager for our chemistry department there, which was very eye-opening and a lot of fun and interesting things working with students in a lab setting. Goodness.
All right. All right, so we're going to talk today focusing on GoDirect sensors. And GoDirect sensors have a lot of advantages. Some of you that do, if you have familiarity with Vernier, you may be more familiar with our older style sensors that plug into an interface. The beauty of GoDirect sensors is that it's just the sensor, does not require an interface. They can either connect to a computer via USB cable or via Bluetooth. So these are fairly versatile. We have a lot of the sensors have improvements over the older models. Some of them have extra channels that we will show you how to access. So generally, these are kind of I would say, a big improvement over the older style. They are easy to calibrate. The menu is in the software, and you can use them with LabQuest. So if you do like working with LabQuest 2 or LabQuest 3 platform, you can connect these sensors again via USB or Bluetooth directly to a LabQuest, as long as it's a LabQuest 2 that has Bluetooth built in, or LabQuest 3. And they also work with a software called graphical analysis, and we'll spend a little time on graphical analysis. The wonderful news about this is that the basic version of graphical analysis is free, and you can get it for 5 different platforms. So if you have a bring your own device situation with your students on your campus, those students can access the software for free on Windows, Mac, Chromebook, or iOS devices, or even Android devices. And we have a link on our website that takes students to whatever platform they need. They just choose the correct one for their device. The other advantage is that the software lives also as a progressive web app, where as long as you are in the Chrome browser, and I believe the other browser that works with it is the Edge browser, but you can just simply navigate now in the browser to graphicalanalysis.app, and it will pull up a session of the software for you. So you don't even have to download something as long as you have a Chrome browser. So I'm going to demo that really quickly just to show you how easy it is to access. It will launch right here in the browser, and it will give you the menu that you use to connect sensors. So it's extremely easy to use and very accessible for your students. They don't need to sign up for an account. They don't need to do anything special to access it. It's just available to them for free. And it does not collect any student information. All right. So graphical analysis, as I mentioned, works with the GoDirect sensors. You can also plug in the older sensors into an interface and connect it to a computer by USB if you have older Vernier sensors. So you can also use a mix of sensors in a graphical analysis session. So you're not limited in graphical analysis to only using the newer style of sensors. You can use your older ones as well, but we will focus on the using graphical analysis with the GoDirect sensors. The sensors that we're going to highlight today are going to include the gas pressure sensor, also GoDirect CO2 gas, and I'll do a quick demonstration of using the GoDirect CO2 gas sensor. Also GoDirect O2 gas sensor, and the GoDirect optical dissolved oxygen sensor. So we'll spend a little bit of time on each one of these and how they are useful for a typical Biology 101 type course. So we'll start with CO2. This one is my favorite. I used this one a lot when I was teaching. This sensor is really kind of a workhorse for teaching concepts like cellular respiration and photosynthesis, because it is measuring gaseous CO2 in a chamber, and it's very easy to use. You can do experiments with yeast metabolism with it. I know that we had a question about splashing, if you splash yeast into a gas sensor. Remember that these are gas sensors, so you don't want them to get liquid on them or in them, if at all possible. If they do get wet, you want to dry it out right away, turn it off, power it down, let it dry. We have had incidences where, for instance, earthworm crawled inside it. That was possibly our most exciting kind of humorous repair ever, was seeing like where an earthworm had climbed into a sensor. But people do use these in chambers with animals, with things like insects and earthworms, other kinds of arthropods. So you can use them for animal respiration, also for soil respiration. If you are teaching any kind of soil science or covering soil science, it's a great way to demo all of the microbial action that happens in soil, to put soil either into a chamber and then put the CO2 sensor in, or you can take a Nalgene bottle, cut the bottom off, and make a chamber that nestles right into the soil. And we can talk a little bit more about that if anybody is interested. So what I'm going to demonstrate is using the CO2 gas sensor for looking at germination of some peas. So I've got some peas that I've soaked, and I'm going to show you here. I need to stop sharing for a second. I'm going to change my video camera over briefly. So I have a CO2 gas sensor and also this chamber with some peas that I soaked earlier today to the just a couple of hours, and then drained the water off. So they've already taken on enough water in that beginning process of imbibition, where they start absorbing water and then they start liberating sugars. And we'll go back here to our sharing. And we'll connect. We'll go ahead and connect a CO2 sensor. So we're going to turn that sensor on. And The Go Direct sensors, when you first turn them on, they will blink red, which means that they are looking for a connection. And remember, I'm using the Progressive Web app right now. We're going to tell it that we're going to make a wireless connection. And then we just have to find it in the menu. I had it paired earlier, so it remembers that it was paired with that sensor, but we're still going to tell it pair for right now. And now our sensor is connected. I mentioned on that earlier slide that the sensors, some of the sensors have extra channels. So this sensor does have a relative humidity channel built in, as well as a temperature channel. They're not on by default, but you can turn them on if you like in software. For now, I'm just going to leave on CO2, but this is how you would enable other channels on sensors. All right. I'm going to slide the sensor into our Nalgene bottle, and that's just a 250 milliliter Nalgene bottle. These sensors are sized to fit the neck of that kind of that standard lab bottle size. And I'm going to go ahead and hit collect because I know those peas have been cranking out some CO2. I'm going to zoom to the data so it's a little bit easier to visualize, but you can see it plotting a sample once every 2 seconds. So as you can see, not a lot of prep for your students to do. They launch the app, they connect a sensor, and they can be collecting data incredibly quickly in lab. And this is particularly nice for students to be able to visualize phenomenon that they otherwise may not be able to see in a concrete sense. We, of course, also have, as I mentioned, the O2 gas sensor. You could use it in this type of scenario as well. Do we have any questions at this point over anything like using graphical analysis or?
So one quick question is, what about any sort of analysis that you can do in graphical analysis? Are there
Yeah,
things
the
like being
name.
able to look? Yeah.
The name indicates that there's some analysis features. Yeah, let me go ahead and stop data collection. So one of the things that we especially like to do in biology classes is look at relationships between parameters with maybe how quickly something happens. So one of the really robust analyses here is being able to fit curve lines or look at the, for instance, the slope of a line as an indication of how quickly things are happening. So we can apply a curve fit to these data, and we can get a slope equation so that students could compare, for instance, multiple samples and see if the slope changes. And you can have them manipulate parameters, for instance, try different types of seeds or different amounts of seeds, or soaking them for different amounts of time to look at differences in metabolic activity. We have a variety of activities, lab activities that we can talk about in a little while, where to find those. But this is where you would find the analysis features. And I realize I did that fairly quickly, but I'll X out and show you again. When you have data on your screen after you've collected it, you can simply highlight and you get a pop-up menu to show you kind of a shortcut to the options for those data. Some of the features, some of the more advanced analysis, things like histograms, we don't use FFTs a whole lot in biology, but those types of features live in the pro version of the app. But the more basic analyses are all part of the graphical analysis app, in the basic app. The other place to find the list, the full list of options, is to click on, in the lower left corner, there's a shape of a graph, a little icon of a graph, and that is where you would also bring up that menu. So if you have X'd out of something and you're not sure where to find it, you can always find all of those options underneath the graph button in the lower left corner of your window. To navigate around, I'll show you a little bit, if you're new to graphical analysis, we automatically get a graph screen when we launch. But what if you want to look at your data table? You've got options here under this in the upper right corner. Looks like a stack of boxes. Those are your view options. And you can turn on or off the data table. So you can toggle it on or off. You can also, if you want to bring up a live meter, you can do that as well. And then to drop that menu, just click anywhere outside of it and the menu will go away. If you would like to rename data sets, you can also do that in graphical analysis. Simply click on the three buttons next to the default name. The default name is always dataset 1, dataset 2, subsequently as you collect them. So you can create your own name for this and simply rename. And now if you click over on the y-axis label, it will pull up your options for what are displayed on the graph. Anytime you have more than one data run, by default, it picks the newest run to display. But this is where you would also find, if you wanted to overlay two graphs, you would simply click on the y-axis label and you could bring up the other options to apply different runs of data, different data sets to the graph. All right. So any questions on our CO2 sensor? If not, we can move on. We mentioned that we can also use this one for photosynthesis. Oh, question. Looks like we have a question. So using older sensors. So how would you go about using graphical analysis with older sensors? You could plug in a LabQuest family interface or a GoLink. For most of our sensors, you can also use a GoLink. And both of those options have a USB cable. So even something like LabQuest Original they all have a cable with a USB-A connection that could be plugged into a computer. So you would plug in your sensor into the LabQuest and then plug the LabQuest into the computer, launch graphical analysis, and the LabQuest will go into interface mode and you could use it in a session of graphical analysis. So the sensors that previously used Logger Pro should, as long as they are sensors that are new enough to auto ID, and I'm trying to remember what year auto ID came about. I want to say it was, Colleen, do you remember? Was it like 2005?
I think it was around 2002, 2004.
Okay.
So
okay.
pretty old.
Yeah, if you plug your sensor into a LabQuest and your LabQuest knows what it is automatically without having to manually set up the channel, then they will be usable with graphical analysis. The only products that don't are things like the really old Lab Pro that looks kind of like a green calculator with no buttons. Those are too old to work with graphical analysis. But all of anything that is part of the LabQuest family of interfaces will work, as well as GoLink, which is a connection for a single sensor. All right, so I realize I did our live demo there with our CO2 gas. So we can kind of move on and talk about a few other sensors, but at some point, if we decide that we want to go back and demo something, we certainly can. But the next sensor that I wanted to talk about is the GoDirect optical dissolved oxygen sensor. This sensor is fantastic if you want to work in an aquatic environment. So if you are working, if you want to either measure photosynthesis happening with algae or aquatic plants, this is a fantastic sensor to do that with. You can also look at respiration in a liquid environment. It is made for aqueous solutions, so you wouldn't want to use this in any type of heavy viscous material or oil. But it is fantastic you can use it in things like fish tanks if you're working in the classroom, or water samples that your students take. You can also take it outside, certainly, and use it. It is splash proof. I believe it can be underwater. Colleen, is it for 30 minutes? As long as the USB is not plugged in, correct?
Correct. Yeah, the whole sensor body is rated at 1 meter for 30 minutes. But once you go underwater, you're going to lose the Bluetooth signal anyways. So it's one of those I don't recommend necessarily doing it yet. But the tip port of the probe that does the actual reading, I've submerged that and left it submerged for 3 months and have had no problem.
And there's that sensor. I brought one with me. It comes with a storage bottle, and the storage bottle just has a wet sponge in it that keeps the end of the sensor damp, but it doesn't have to be stored in water. It just needs to be kept humid. But yeah, this is a fantastic sensor. If your students do any type of field work, if you take them out to do ecology experiments or environmental science experiments, or watershed testing, this sensor is just absolutely fantastic. It has built-in channels for temperature as well as barometric pressure, and you can capture concentration as well as saturation of your water. So you can show the relationship between temperature and oxygen content of your water. And Colleen, feel free to jump in at any time here if I forget anything with this sensor. So this is an example of doing something like biological oxygen demand using this sensor. Let's see. Oh, and we also have, I forgot, we have a salinity corrected channel. So if you are working in an estuary or in saltwater, you can put in your known salinity and it will automatically correct the data for you.
Exactly. The big key to remember is that it's salinity correction. It does not actually measure the salinity. We have a separate salinity probe for that, and you just have to tell it what the salinity is of the solution you're in.
Right. So the main calibration is for freshwater. That's what realize most educators will be probably working in freshwater, but we do for anybody that's on a coastline, we do have that special channel for correcting data for salinity. So you would want to just measure that, as Colleen mentioned, separately. All right. From there, we can move on to gaseous oxygen. So we have the oxygen gas sensor, and I'm gonna lay it down, which we don't normally do. But the oxygen gas sensor, normally we remind everybody all the time, keep it upright. It does have an electrochemical gel inside. It has it works by basically gradually breaking down that gel in the electrochemical cell. So it does, usually about after 6 to 7 years, it does need to have that cell replaced. But this is incredibly useful, much like CO2. It's great for looking at photosynthesis as well as cellular respiration. But it's also great for looking at enzymatic reactions that produce oxygen as a byproduct. So looking at something like catalase reacting with hydrogen peroxide, this is a great sensor for doing that. We often, a lot of educators use this in concert with CO2 so that they can show the relationship during photosynthesis or cellular respiration of those two gases both changing at the same time. So I'm trying to think of anything else with this one. This one also does have a channel for temperature. So you can actually get the temperature inside your chamber in addition to the oxygen gas reading. All right. From there, we can move on. Unless we have questions. Oh, can you replace that cell?
Yes.
So the, yes.
Sorry, I was just about to jump in there of like, yeah, so the cell in this one's just like the cell in the current LabQuest version, and it does need to be replaced every 6 to 8 years, which is nice because then you don't have to purchase an entirely new sensor.
You set up a repair with us and send it in-house, and there's a fee for it because it's about half the cost of a new sensor to replace the cell, but then it should give you another 6 to 7 years. So, and with the oxygen gas, we do recommend calibrating it maybe once every 6 to 12 months, just to keep it, as that gel breaks down, the reading will drop a little bit, but if you recalibrate it periodically, it should give you good readings up until the point that the cell is no longer has enough voltage to it. Right. So next we have, this one might seem like an odd choice for a biology webinar, but it's the gas pressure sensor. And this sensor can actually be used for things like grip strength experiments. If we have an accessory for it, that's a squeeze bulb. This sensor comes with an accessory kit, and that's actually what's depicted in the shared slide. The image has some of those, some of the tubing that comes with this sensor. And it's being used in the image for an enzymatic reaction between hydrogen peroxide and catalase from yeast that is producing oxygen gas as a byproduct. So instead of measuring the oxygen gas itself, you can simply measure the pressure change. And this is a very robust prep. You can have students manipulate things like the temperature of it, or the amount of substrate or the amount of enzyme. But it's a very fun lab for them to do. But you can also connect this tubing, for instance, the sensor has a lure lock end on it, and the tubing that comes with it, you can connect it to a squeeze bulb if you'd like to use it for a simple experiment with grip strength. Or we have a prep that actually can be connected to a cut stem of through the tubing. You flood the tubing, other than you leave a bubble outside the sensor itself, but you can connect it on the other end to cutting from a plant and look at how quickly water moves through plant material. So if you do any kind of a section on plant anatomy, or how transpiration moves water through the xylem tissue of plants, it's a great way to demonstrate how much plants move water from groundwater back into the atmosphere. So I used to do that lab when students were working on ecology and understanding the role of trees in the environment in moving humidity back into the environment. You can also do fermentation studies with it. So again, using pressure, in this case, CO2 gas produced from fermenting yeast. But this is definitely kind of a workhorse sensor for life sciences. Colleen, can you think of any other comments on this one?
It's one of those that I like to point out that depending on kind of the budget cycle and what budgets are, is to me, this sensor is a cheaper version of a sensor that can do a lot of different things. So in my opinion, you get a lot of bang for your buck with it, because of this versatility where, there's like enzyme action and fermentation. Yeah, we could do those with either an oxygen gas sensor or a CO2 gas sensor, but it's kind of nice to have this gas pressure sensor because of how versatile it is. And it also makes it, the data is, because it's in a smaller volume, tends to be a lot quicker of a data turnaround, so that lab time actually is essentially being used a lot more efficiently.
That's a great point. Yeah, we often do this prep at workshops, and it makes for a really easy workshop experience where participants get data so quickly that they can then analyze and manipulate, that it's just a really robust prep. All right. Well, the last thing that we were going to highlight was actually an instrument, and that is our GoDirect SpectraVis Plus. And I'm not going to demo it, but to tell you a little bit about it, we do have a variety of spectrometers. The GoDirect SpectraVis Plus is really ideal for a biology course. It's used a lot also in freshman chemistry courses. The wavelength range is just barely into the UV, so 380 all the way up through the visible spectrum, all the way to 950 nanometers. And it also works on a free app. So the app for spectrometers is going to be spectral analysis, much like graphical analysis that I showed you. You can either download it on those different platforms. The same 5 platforms are available as for graphical analysis, or you can run it right in the app as a progressive web app. You just type in spectralanalysis.app and it will launch and you can connect a spectrometer. And this is fantastic for any type of analysis of plant pigments, especially chlorophyll, but you can also look at other photopigments. You can also use, it has a fluorescence mode where you can hit the chlorophyll extract with light and have it fluoresce bright red. So you can do that if you've ever done the bloody chlorophyll experiment with students. You can use that to demonstrate the fluorescence of chlorophyll. It's also great for enzyme studies. We have a number of enzyme labs where you can use this to look at enzymatic reactions. We also have a lab where you can look at it uses chemical DPIP, where you take a chlorophyll extract, where you're actually extracting the chloroplasts, and you can see essentially electron transfer to the dye, because the dye will change color. So you can see with different amounts of light or different timing, the change in the color of the dye as an indicator that those chloroplasts are still working, basically, still going through photosynthesis. If you are interested in spectrometers, I did drop a link in that will take you to a great webinar that we did that goes through all of the different spectrometers that we offer. So if you do spectroscopy in your lab, I do recommend checking this out. It will walk you through all of the different features of the different spectrometers that we have. We do have some spectrometers that are capable of doing things like DNA quantification. The SpectraVis Plus is not, the GoDirect SpectraVis Plus is not capable of that, but we do have some spectrometers that are. So. All right. So if we don't have any other questions about sensors, we can talk a little bit about what labs we already have available for these different products. We have a collection of lab books, and I highlighted the 4 main lab books here. For biology, we generally recommend this Biology with Vernier book, which is more of a cookbook style. So if you want your students to kind of have a set of lab instructions that are not that they're rigid, but that it's very stepwise. And that one is that one has a collection of 31 labs, and many of those labs have different versions in the books of different options for different combinations of sensors. If you prefer a more open-ended approach where students are prompted to design their own experiment, we recommend this Investigating Biology Through Inquiry book. That book is much more open-ended. There is guided inquiry as well as open inquiry options. So actually, when I was teaching college, I used a combination where early in the semester, I would have my freshman biology students start just to learn how the sensors work. I would have them start with labs from Biology with Vernier, and then I would transition them once they were a little bit more comfortable with scientific method and designing experiments. I would move them to labs from Investigating Biology Through Inquiry. And it was my students really responded wonderfully when I started letting them design their own experiments. Just to mention these other 2, we do have human physiology as well. So if you teach human physiology, we have a full line of sensors for that topic as well. And the way that our lab books work is your department only needs to purchase it once. So some of you may actually already own them. The way to find out is if you create a Vernier web account, use your college or university email address, because sometimes it will automatically tie you to any resources. But it also makes it easier for us to find your resources if it doesn't automatically connect in your account. Make sure you tell us what department you're in, because the book licenses go with the department. So even if somebody bought it 10 years ago and moved on to another job, your department, if you ever bought it, they still own the license, and that license can be accessed by anybody, any of the faculty or staff in that department. So I do encourage, if you don't already have a Vernier web account, set one up, because it's a great way to find if you already have some resources available to you. All right, it looks like we have a question. Oh, Colleen, did you get that one?
Yes, I got the question was about LabQuest sensors, if the oxygen LabQuest sensors could have their cells replaced as well. And the answer is yes, as long as they aren't too old. There's a certain age limit, and I can't remember, somewhere between 2007, 2000.
Yeah, we do have a TIL on it. We have a Tech Info Library article that has, it helps you identify the body style of the sensor that can have the cell replaced. I just don't remember the year off the top of my head, but it is right in there. It's like, I think it's like mid 2007 or something is when.
Yeah, it's a very specific time when, and it's basically a form factor change. And we have like pictures and everything on there to help guide you, to let you know how old your sensor is.
And you can always, if you have any trouble finding it, you can always drop us an email, biology@vernier.com, and we can help you find it.
Mm-hmm.
All right. So I'm going to move on to just talk a little bit about what we have online as resources that you can access. We do have a whole portion of our website devoted to educator training. So when you go to our website, right across the top bar, there are different buttons. If you hit the educator training button that's circled there, it will pull up several other tabs, including one that goes to our webinars. So like this one being recorded, all of our other ones have been recorded and are online. You can also request personalized training, and we can do up to an hour for free with you through a Zoom session. But if you want something longer, you can fill out that form, request it, and we can get back to you. We also have some information for if you're looking for grant money. We have some tips on writing award applications, as well as we have links to videos, tips and tricks videos, short videos. We also have a number of white papers that are also handy for if you're writing grants to get new equipment for your lab, as well as links to things like product manuals. I also wanted to mention, we have a YouTube channel that is fantastic. If you have trouble finding specific videos on products that you're looking for on our main website, try our YouTube channel, because our YouTube channel goes back multiple, like we have videos that go back several decades. And so if you have older equipment, chances are there's a video on it on that YouTube channel. And then I mentioned earlier the personalized training request form. I just put a hot link into it in the presentation, in case anybody wants to get to that quickly. But it's just basically you fill out this very simple form, tell us what kind of training you need, and then we get matched up. You get matched up with one of us to discuss training. All right. And Dylan, this is Dylan's part.
That's right. So we're doing a giveaway, and the fact that you are still here and enjoyed this webinar means that you have been entered to win your choice of a GoDirect pH sensor or a GoDirect gas pressure sensor. So I entered everyone's names into a random name generator, and today's winner is William P. So congratulations, William. I'm going to go ahead and email you. I'll just need to know what sensor you'd like us to send and some shipping information. So keep an eye out for an email from someone with an @Vernier email address and reply back to me, and we'll get that shipped out to you.
Fantastic. Congratulations, William. All right, the personalized training link. If you are on our website, so this is just like a screen capture of the main web page. If you navigate across here to educator training, it brings up this little drop-down menu that's shown on the slide. And you would go to that second tab over from the left that says personalized training. That will take you straight to that page where you can schedule a training with us. I did link it also in the presentation. So if you end up getting the slides, if you wanted to download the slides, you can also access it through this. Nice to see you too, Judy. All right. So if you are left with any lingering questions, or if you think of questions later, you can always reach us. We have a couple of different channels that you can reach us on. We do monitor email. It's usually going to be Colleen or myself that are going to be answering your emails. They go to biology@vernier.com. We also have live chat on the website during kind of West Coast business hours. We are in Oregon, but our chat does come on at about 6:30 in the morning. I mean, you can put a question in the chat before then, but you might not get a human quite yet. But we do start answering. I'm one of the early morning people. So if you happen to be on the East Coast and you call in the morning or send us a chat, most likely you're going to get me. Later in the day, you're more likely to get Colleen. But it's pretty much usually the two of us on the biology side. You can also fill out a form on our website. You can fill out a help form, and that will create like an email chain that we can contact you. So whatever mode of communication you're most comfortable with, you can find us. So, all right. Thank you so much for all of you attending. We really appreciate it, and we hope that we can help you with any questions that you might have.
Absolutely. Bye,
Thank you
everyone.
all.
Thank you, everybody.